That's Dr. Fillmore from psychology. All right, go ahead and fire up top hat. So we're in the process, we're converting everything from CD to digital. So I've got a couple more. Sorry about Tuesday. Braces popped open, tore up his gums, and I had to skip class. But he's fine. He's a 6'2" weenie. Thank you. All right, we top hatted. Wait a few minutes for everybody else. Exam 3 will not be 2. It will not be moved. It'll still be next Thursday. I'm still trying to work on what's going on with the Zoom recordings. Luckily, Jason's here recording, so anything between my Zooms and Jason's recordings at work, we'll get you access to it tonight, tomorrow, no later than tomorrow, hopefully. Steal more. Oh, did I cut it? Oh, I screwed it up, sorry. Okay, fire top hat. Get yourself a good one. Tuesday will be an exam 2 review. And I will put a study guide up, but again, the big thing here with this, know your enzymes, right? No chemical structures, as in NO, not KNOW. You don't have to know any kind of. You have to know chemical structures, the rest of the class does not. Faith has to know chemical. No. Do not study chemical structures. This is not a chemistry class. We're not going to do it. Okay, I'm going to start top hat now. Know your enzymes. We didn't do this already, did we? Acetylcholine questions? No. Okay. New ones. Yeah, I like the small ones too.
She can log in the top hat. Like always.
Like always. What's the problem?
She's not in top hat.
Keep trying to get in. Good. Acetylcholine esterase. Good job. Is anybody else having trouble with top hat? Is anybody else having trouble with top hat? Is it? Everybody else is okay. Now remember, so that you can use the in-class quizzes to study for exam 2. I'll make them all available to you after class today. Complete guess? Is that what I heard? Fair. I forgot to add 60 seconds. Okay, we're almost there. 108 over 112. Let's see how you did. This is a tough question. Okay, good job. Yeah, the alpha 4 beta 2. We talked about this for about 5 seconds, so don't freak out about that question. Which acetylcholine receptor responsible for the hallucinogenic effects. Let me usually think of these two. Yeah, they turned out.
Nice. I love that tree. I love it.
Go. What happened? 5 seconds. Good, muscarinic receptor. Stimulating muscarinic acetylcholine receptors at a high level can cause hallucinations. Blocking muscarinic acetylcholine receptors, blocking them, reduces nausea, which I should have used last time I went fishing in the Atlantic side of Florida, because I chummed the water for 4 hours.
What's the weak note that does it?
Scopolamine. This should be an easy one, hopefully. I'm sorry, what? Oh, Lauren's driving somewhere. Okay, I'll go get on the Zoom. I didn't know, she's on the road for something somewhere. Well, where's my Zoom meeting? Oh, there it is. All right, where do I find my waiting room? Anybody? Where's my waiting room on Zoom?
She hasn't requested. She does, then it'll show.
Okay.
Click the participants.
Say what? Who? What?
Click participant. No.
I didn't mean to do that.
No, keep going the other way. Left. There
Participants?
You go. Yeah. And then click it, and then wait. It'll show up.
Yeah, there's nobody there. Okay. Yeah, there's nobody there. Okay. Let's see how you done on this question. All right, good. Hippocampus. I think that's the last one. Yeah, that was the last one. Okay. One more thing about hold on. I'm not going to teach without my big blue dot of science. Yes, we're on amino acids. All right, so there won't be any new material on Tuesday. Tuesday will only be a review. You get my pointer set up. I don't have to do this every time. Oh, come on. Okay, there we go. Okay, we're good to go. Okay, so top hat questions will be available tonight or tomorrow morning for you to review. If you missed a class with an excused absence and didn't complete an in-class top hat, send me an email. I want to make sure you get those free tiny slivers of extra credit points. Again, Tuesday will be review for exam 2. Now we're in midterm now, so all of your grades from top hat after exam 2 will be shifted to Canvas 2. So you'll be able to see exactly where you are at midterm. You can see exactly where you are on top hat 2. Now, if the class as a whole is doing great. If you do get an academic alert about where you're sitting at in Psych 459, you know the process. That's just me saying, hey, let's talk about how to study for 459. I don't know if that's going to happen or not. So let's start with our last. Oh, don't do this to me. Oh, jeez. $95. This thing. It's the Cadillac of pointers. In feel stubborn today, so I'm not going to teach until I get this figured out. We only have like 16 slides to go. This isn't a UK, this is a Logitech presentation software that usually works just fine. I love having an IT guy. He helped me in Psych 302, or Bio 302. Oh, there we go, that's it. That's it, that's good. So it should work. Yeah, that's what I need to do. Oh, I've got to have the PowerPoint open. There we go. Thank you. I don't know what he did, but he did it. Okay, so last neurotransmitter system we're going to talk about today, okay? So after exam 2, we start diseases or classes of drugs, abuse, psychotropic meds, and so forth. So you get to apply everything you've learned on exam 1 and 2 to actual therapeutic conditions, which is the fun part of this class. Everything we've talked about so far are characterized as small molecule amino.
Oh, you might need to get closer to the computer itself.
I should be on. Okay. Small molecule neurotransmitters. Okay? You need a very small instruction. We're not going to go to large molecule. Luckily, we only have 16 slides, so today's fiasco will be over. Okay, so what did you notice when we talked about monoamines, including dopamine, norepinephrine, serotonin, and acetylcholine? What did you notice about where they're made? Okay, Jason's getting there. Are they made everywhere throughout the central nervous system? No. Everything we've talked about so far, they're made in small clusters of nuclei, small clusters of neurons, in the basal forebrain or in the brain midbrain, and then they send axons all over the central nervous system, all over the brain spinal cord. Okay? What we're going to talk about today is small amino acids that act as neurotransmitters themselves. What do I mean by small? Well, this is, maybe I'll just stand right here. Those are two very small amino acids. Again, you don't need to know structure. They're very small molecules, but they're very, very important when they act as neurotransmitters. Now, large molecule neurotransmitters, we're going to talk about a little bit, for example, when we talk about opiates, the molecules that our central nervous system make that stimulates opiate receptors are very, very large molecules. They look much more like this, okay? But today we're just going to do small molecules. All amino acids, again, this is just a little bit of basic chemistry. All amino acids share a very common molecular structure. They have an amine group. Okay, maybe, do I have to be over here? Nope. I give up. Amine group, NH2, and negative COOH, which is a carboxyl group. Amino acids vary only in a tiny little side chain. That's not an important point, but it's interesting because a really small chain in that R makes the amino acid work as an entirely different neurotransmitter. And that's what we're going to go through today. It's a pretty cool thing. Now, you know amino acids is the building blocks of protein, right? Muscle and other proteins. But a couple of them act as neurotransmitters themselves. We're just going to talk about the two most common. That's gamma-aminobutyric acid, or GABA, right? You've heard of GABA before? GABA is the primary, most abundant inhibitory neurotransmitter in the central nervous system. Glutamate is the other one we're going to talk about today. The most widely produced excitatory neurotransmitter in the central nervous system. Okay? Oh, jeez. This thing is going crazy today. But there are other amino acids that work as neurotransmitters. You're only responsible for knowing GABA and glutamate, okay? So what's really different about GABA and glutamate compared to dopamine, norepinephrine, serotonin, GABA and glutamate are made in every single region of the brain and spinal cord. They're made everywhere. They're the only two neurotransmitters that are made everywhere. Okay? That tells you a couple of really important things, right? One, GABA and glutamate are extraordinarily important for central nervous system function and drug action. Why else would we have evolved to make GABA and glutamate everywhere, from the bottom of our spinal cord all the way up to our prefrontal cortex? They're involved in all neuronal activity. One's excitatory, one's inhibitory. So I want you to think a little bit. I know it's Thursday. Everybody wants to meet inside. Why do you think we have an excitatory and an inhibitory neurotransmitter that are made everywhere in the central nervous system right next to each other? Just think about that from a biological perspective. Anybody want to take a shot? Yeah, Esteban?
Homeostasis?
Okay, homeostasis. Isaac, do you want to expand on that? Yeah, Eliana? Okay. I brought two squishy groups. Yeah, homeostasis. So we talked before about the action potential, how we keep neurons at like minus 70, really close to firing, right? Because biologically, we're animals. We need to be ready to fight or flee. GABA and glutamate are always opposing each other to keep the neurons in our central nervous system at rest until a significant stimulus occurs. When something significant happens, might be a sound, a smell, waking up in the morning, glutamate goes up and wins the fight. But on a millisecond by millisecond, GABA keeps neurons quiet. It's the second way that neurons are kept at minus 70 quiet, GABA. Does anybody know what are the four ions we talked about? GABA, the main GABA receptor that's inside of neurons when it's activated. It's inhibitory. We've only talked about one inhibitory ion all semester. Chloride. Cl minus. The most abundant GABA receptor, when it's bound by GABA, opens up with chloride in. What does that do to minus 70? Takes it to minus 75, to minus 80. So guess what alcohol does to that GABA receptor? Opens it. That's why the action potential is so important. Sometimes the answer about how a drug works is just that it opens or closes a neurotransmitter receptor. Okay? Glutamate does a million different things. About 70%, let me see a long stick or something in here I could use. Some classrooms have that. A classroom in Morgan has it. About 70% of all neurons in the central nervous system use either GABA or glutamate. Another example of those neurotransmitters being so extremely important for millisecond by millisecond neuronal activity or inactivity, okay? They have both inotropic and metabotropic receptors. Now here's, just like the other neurotransmitters, what I want you to remember is the enzymes, not the chemical structures. Okay? Glutamine is a simple amino acid that our bodies can make. This is where I really need a pointer. Glutaminase, if I make glutamate, I'm also a neuron that makes glutaminase. I have the genetic code to make glutaminase from DNA, then messenger RNA, and so on and so forth. Okay? Glutaminase leaves that amine group on the far right of glutamine off. That makes the active excitatory neurotransmitter glutamate. Oh, this is really bumming me out. I don't have anything here. I know. Okay.
Are you using the other end of the pointer?
No, I shouldn't throw squishy right now. Yeah. But again, so that's why I do this kind of stuff, okay? What's missing? It's that NH2, the amine group. Simple organic modification of glutamine makes the most excitatory neurotransmitter in the central nervous system. So know that. Glutaminase says, hey, I make glutamate. That says I'm a neuron that makes glutamate. Okay? Jeez. This looks really, really scary. It's going to be okay. Deep breath. It's gonna be happy. Oh, that's right. You saw this last fall.
Wasn't this like the last exam for
Yep. Intro to neuro, yeah. That's how important it is. You need, if you're taking classes like this, you need to know about glutamate and GABA. Two most critical neurotransmitters in the central nervous system. About 50% of you are pre-med. You gotta know this stuff by the time you walk. You cannot walk in graduation next May if you don't know how glutamate and GABA. Okay? The way to remember this kind of stuff is to go with the cookbook, the recipe type of study, right? 1, 2, 3, 4, 5. Step 1. Huh? Somebody ridiculing my digital mastery? You have the animation too on camera. I've been teaching this a long time. This is the way you are going to learn best, I promise. This is how you're going to learn best. I study pedagogy a lot, how to teach people STEM. This is going to work. So if I'm a neuron that makes glutamate, I have, oh, don't move. Shh. Scared to hell. If I'm a neuron that makes glutamate, I have a transporter on my nerve terminal that transports glutamine into the neuron. That's step 1. Step 2, I also make glutaminase, which cleaves off that amine group off of glutamine. Now I have glutamate. Step 3, glutamate. Put that away. Glutamate gets transported up into synaptic vesicle to await an action potential to get released. Okay? 1, 2, 3. Now you know how the synthetic pathway. Now if you take Biochem 401G or something, you're going to need to know structure. But you don't need to know it for this class. So what's very different again about amino acids that are neurotransmitters? What happens after the neurotransmitters we talked about before? What happens after they get released? They go bind to a receptor on neuron number 2, then they detach. What happens next? They get reuptaken, right? Glutamate and GABA don't get reuptaken very much. About 20% of glutamate and GABA, we'll talk about GABA in a second, get reuptaken. I will settle for an arrow if that's all I get to. Okay? About 20% of glutamate in this example gets reuptaken into the neuron that just released it. Only 20% or so. Oh, too far. 80% of glutamate that's released acts at a receptor, then pops off, gets transported at step number 4 here into a nearby astrocyte. Remember astrocytes from blood-brain barrier? Right? You have that little foot out there plugging a hole in the blood-brain barrier. Astrocytes do a million things to help neurons function adequately. EAAT just means excitatory amino acid transporter. It's a transporter that uptakes excitatory amino acids like glutamate. We'll talk about why most of the glutamate doesn't get reuptaken into the neuron that just released it. Why does it have to go get reuptaken, not reuptaken, get uptaken by a nearby astrocyte? Then glutamine synthetase catalyzes a reaction that adds that amine group back, producing glutamine. Then the astrocyte ejects glutamine into the extracellular space to start process at number 1 again. Really efficient 6-step process. I mean, the biochemistry is a lot more complex, but for this class, this is what we need. This is all we need to go through. You'll be ready when you get into Biochem 401G to deal with the more intense biochemical aspects of it. Questions about that? When you're studying this, have this slide up there, have a blank sheet of paper nearby. Step 1, glutamine uptake. Step 2, well, I think that's the best way to study this. This is just me recommending how to do it. Yeah, Theo?
Is there like any advantage, like reuptaking glutamine and redoing the whole process instead of like reuptaking glutamate?
Yes.
Right?
Yeah. I don't remember if I included it on this slide. I did. Now, there's a big red, deep breath. There's a big red X on this slide. Why does 80% of glutamate need to be uptaken by an astrocyte instead of the neuron that just releases it, okay? This is the Krebs cycle. I get it. There's a big X on there. You don't see it? Gonna come walk us through it? Okay? So those of you that have had biochemistry, what do glutamate and GABA have to do with the Krebs cycle? Mitochondrial energy production. How neurons produce energy. Glutamate and GABA are substrates, meaning they're used to make substances in the Krebs cycle. Glutamate and GABA do more than, what's that? What are you talking about this? Oh, God. I don't know. It's worked all semester so far. I think I need to reboot the computer. But I'm not going to do that today. Okay? So glutamate and GABA, it turns out, the two amino acid neurotransmitters we're talking about today, they're also heavily involved in the Krebs cycle. If I overload a neuron by reuptaking 100% of glutamate and GABA that's free floating through the cell, the cytosol, I'm going to overload the Krebs cycle at some stage. Does that make sense? What stage doesn't matter. We'll talk about that when we take Biochem 401G. So it's more healthy. It's healthier for a neuron to send most of the glutamate back to an astrocyte to be broken down to glutamine, rather than just reuptaking all that glutamate and having excessive substrate in the Krebs cycle. It's unhealthy for mitochondria to reuptake 100% of the glutamate you've just released, because glutamate does a million things, and one of them helps with mitochondrial energy production. Kennedy? Mm-hmm. You're not going to get any questions about the Krebs cycle. That's just kind of a Jeopardy level thing, okay? The two main glutamate receptors we talk about are the NMDA, N-methyl D-aspartic acid receptor, and the AMPA receptor. These receptors are named simply for human-made drugs that bind to them and activate them. The NMDA receptor is an ionotropic receptor that allows calcium, sodium, and some potassium to flow into neurons when it's activated. So what effect is that going to have on resting membrane potential? It's going to depolarize it, right? Ca2+, Na+, K+. You're going to flow into a neuron and quickly change the membrane potential to be more excitatory. An AMPA receptor is basically a sodium channel activated by glutamate. So both of these receptor subtypes for glutamate are ionotropic and emit positively charged ions in. That's the primary way that glutamate is the most excitatory neurotransmitter on the planet. Now, I'm going to ask you to think like a neurologist. What would you do if somebody came in and said, we have no drugs to treat seizure disorders, like epilepsy, create a new drug based on this slide? What would you do? Anybody? Ryan? He has a very pensive, studious look on his face. So I thought clearly you had the answer. Welcome to the show.
Hi, thank you for having me. You create an NMDA and AMPA inhibitor?
Yeah, you create drugs. You already have on my blank. You create drugs that would reduce the activity of NMDA and or AMPA receptors, right? Does that make sense? Pharmacology is usually just that simple. Start doing it for you. Okay? Do you know what topiramate is? Anybody ever heard of Topamax? Anybody ever heard of Topamax? It's one of the two most widely prescribed drugs to treat convulsive seizure disorders, like epilepsy and related disorders. Topamax blocks AMPA receptors for a short time. That's it. If you block sodium inflow through AMPA-type glutamate receptors, you can control seizures to a pretty good degree. Very simple pharmacology. Yeah. But no, the question is, what's the primary difference between the NMD and the AMPA receptor? The AMPA receptor is selective for letting sodium flow in when it's active. The NMDA receptor lets calcium, sodium, and potassium flow in. The other most commonly prescribed medication to treat seizure disorders blocks sodium channels, voltage-gated sodium channels at the nodes of NMDA. It just reduces action potential. Yeah, question?
Yeah, you said that Topamax will help with like seizures. I take it for chronic migraines. So does it work the same for that?
Yeah, same AMPA receptor mechanism, but same thing. Migraines are partly an overactivity of the central nervous system. Yeah, so your blood vessels dilate. That stimulates pain receptor or pain pathways, pain axons that carry pain to the spinal cord. Same thing though. You're reducing action potentials by blocking that sodium input. Good. Everybody hear that? It takes Topamax to control migraines, but it's reducing action potentials in your pain pathways. Is it effective?
Yeah, it works.
See? Yeah. Just by knowing a little bit about the action potential and these different receptors, you know how to treat. Now you know everything about how we treat seizure disorders, other disorders like migraines that are characterized by overactivity of central nervous system. Okay? These receptors and regulating them with glutamate is also very important to long-term potentiation and memory formation. You remember LTP from what other previous class you had? Okay? In a lot of brain regions, most brain regions, AMPA receptors and NMDA receptors are buddies, right next to each other, sitting in the neuronal membrane. When glutamate's released, it binds first to an AMPA receptor. It's a lot like Spielberg, isn't it? Special effects, yeah. Okay. I took one design class as an undergrad student at Iowa State. It was pre-Google. Everything was still from the C prompt. That's why I'm so bad at it. Okay? So glutamate binds to an AMPA receptor, blocks or unblocks the receptor. I don't care that you know about polyamines, but at rest receptors, these two receptors are blocked. Now, sodium can flow into the neuron, depolarizing the membrane. When that happens, this membrane here, right between the AMPA and NMDA receptor, is depolarized. The NMDA receptor, which also has glutamate, that's my action potential. Shazam, anybody? Yeah. Now, magnesium, which is blocking an NMDA receptor like the stopper on an old-timey tub drain, that magnesium pops out. Calcium and sodium and a little potassium flow in. Does anybody remember from your prerequisite class what this little bit of calcium does to help with LTP, long-term potentiation, and memory formation? It's an important one. We're going to come back to it a lot. Okay. Keeps it open for longer. What that reflects, this calcium does a lot more than just change the membrane potential. Calcium is the only one of those four ions we talked about a lot that acts as a quote-unquote second messenger. Yes, it helps to change the membrane potential to be more positive, cause action potentials. Calcium also binds to many, many intracellular proteins in neurons to cause the growth of dendrites when it's stimulated at a high level. So long-term potentiation is a calcium-dependent growth of dendrites. That's the biological basis of memory formation. When your brain is stimulated, your hippocampus is stimulated at a very high level. The hippocampus is wired to say, oh, something important is going on. So I'm going to stay excited for several minutes or an hour or two after. Like you're studying for exam 2 the night before, next Wednesday night. Hopefully your long-term potentiation is firing away. But that depends on calcium causing a lot of things to stimulate genes, which help dendrites grow. So your dendrites grow like little tree branches when they're stimulated to the hippocampus. It's a cellular basis of memory formation. Do we remember better or worse when we're intoxicated with ethanol, alcohol? Okay? I wouldn't know. Green tea for me, that's it. Okay? One of the primary reasons alcohol is an amnestic agent, when we get to moderate to high doses, alcohol blocks the NMD receptor, just like the stopper in an old-timey tub. That's why you don't remember things when you get intoxicated. That's simple. Your body could be all eye of the tiger, but your brain is not recording anything. That's it. Now, could I give a drug that overactivates NMDA receptors to help me remember things better? No, you'd have seizures. Remember? You'd have seizures. Questions? Important slide.
The red square is what?
That's where PCP and ketamine bind. I hadn't gotten there yet. So recreational drugs, drugs of alcohol. Ketamine, have you heard of Special K? I don't want to go deep into that now because we're going to do a whole lecture on dissociative and hallucinogenic drugs. Ketamine, Special K. It's a veterinary and a pediatric anesthetic. One of the ways that causes you to not feel pain and not remember surgeries, or horses and babies, because it blocks the NMD receptor. The receptor does a million different things. They also bind down in that channel that lets calcium and sodium and some potassium flow. Alcohol, don't worry about zinc. We're not going to go into it in this class. I'm sure it's okay taking that out. Questions? Really important because we're going to come back to this. Yeah, Eliana. Okay, we're getting into the alcohol lecture, but this is one of the things what my lab studies. So Eliana's question is, does constant alcohol consumption, right, which is about 14% of people 18 and older in the United States, have a quote-unquote alcohol use disorder based on the largest 50,000 person study, does it just continue to inhibit the NMDA receptor so you don't make any new memories? No. We've talked about this in the first exam block. What does an NMDA receptor do? What's a neuron going to do if alcohol is constantly flooding my central nervous system, blocking NMDA receptors so I can't make memories and other things get weird? If this is weeks and months and years. Yeah, Neo has it. Neurons are going to make more of this receptor to try and counteract the effect of continuous alcohol consumption. Does that make sense? That's up-regulation. Neurons are very good about saying, hey, you're inhibiting this receptor too much, I'm going to make more, or you're overactivating this receptor too much, so I'm going to make less.
Can you explain how they're related again? So is the in plus depolarizer regulating
The AMPA receptor has to first, yeah, this is in most brain regions, AMPA and NMDA there. The AMPA receptor has to be activated to allow sodium to flow in, to depolarize this little bit of neuronal membrane between the two. Now, the NMDA receptor says, oh, okay, I'm depolarized right here. Now it's time for me to change my shape, let magnesium pop out. That allows calcium, sodium, a little potassium to flow in.
So does glutamate still bind the
Yeah, glutamate binds both receptors. Yeah, I mean, they're right next to each other. There's glutamate release on it, they're both going to have. But the key is AMPA has to depolarize the membrane next to an NMDA receptor for that NMDA receptor to open up. Yeah, Carter. I'm not going to ask you any questions on the exam about polyamines. Polyamines are like a plug in an old-timey drain bathtub. They prevent sodium from flowing in when that AMPA receptor is not activated. I'm not going to ask you anything about that. I took this slide from a talk I gave at Loyola a couple years ago because I studied polyamines, and you don't need to know any of that. This is really complex. But again, bio, psych, neuro, you need to know why and how NMDA and other glutamate receptors are so important in the central nervous system. Yes, Esteban.
Why do the NMDA receptors have, have glutamate receptors 1 and 2?
Stoichiometry. So we talk about that in Bio 302. Every ionotropic receptor is made of 5 or 6 different subunits that make it more or less sensitive to glutamate. That's it.
The more subunits, the more sensitive?
No, it's not the more subunits. It's which subunit. Is it a 2? Is it a glue 2? Is it a 3? Is it a 4?
So the number's not to differentiate them?
No.
It's more like how sensitive they are.
Yeah. Not every neurotransmitter receptor that has the same general name, like NMDA, is equally as sensitive to glutamate. Some open more slowly, they'll let a little bit of calcium in. Some open fast and stay open for a long time. I'm not going to ask you anything about that. Other questions? This is a really important one. Other questions?
Okay, so what's inhibited is at rest would be the polyamines and magnesium when activated, sodium, calcium, and sodium.
Yes. Haley's got it. We're not going to talk about metabotropic glutamate receptors for the rest of this semester, so you're not responsible for them. Just know that there are a lot of metabotropic receptors for glutamate. Some are stimulatory and some are inhibitory. That's it. How could I ask a question on something that simple? It's not important.
Wait,
No.
what'd you just repeat all that?
We're not going to talk about metabotropic glutamate receptors for the rest of the semester, so I'm not going to ask you anything about that on exam 2. They're very important for a million things, but not for what we're studying this semester. There are not FDA-approved drugs that regulate, moderate metabotropic glutamate receptors in the arena of neuropharmacology. So you're not responsible for but do know, that, like every neurotransmitter, they're both ionotropic and metabotropic receptors. We're almost done. You'll know why we're almost done in a second, because it's pretty cool. Okay? Slight modification to glutamine. Chop off that amine group, the NH2. Now you have the most excitatory neurotransmitter, glutamate. Let's talk about the completely opposite side of neurotransmission, which is gamma-aminobutyric acid, or GABA, the most inhibitory neurotransmitter in the central nervous system. Anybody who doesn't have a squishy brain know what the immediate chemical precursor to GABA is? Locked and loaded? Okay, good. Good job. Good job. Glutamate. Right? Right? Mind blown. Mind blown. One tiny little modification of glutamate turns it into the opposite type of neurotransmitter, the main inhibitory. Again, why do we need, this is just an example. Why do we need, I love the picture, that's why you're here to see. Why do we need inhibitory neurotransmission? Well, this is a big pyramidal cell in purple, and you see all around it, it has smaller cells releasing GABA on it to keep it quiet until there's an important stimulus. So GABA is, I call it the shush of neurotransmitters. It's not yelling, shut up, it's just until something important happens. GABA is keeping neurons quiet, relaxed, super chill, until something important happens. When something important happens, glutamate wakes up this big purple pyramidal cell. So GABA is there to keep the volume down until something important happens. Questions about that? And this happens throughout the central nervous system. GABA is everywhere in the spinal cord in the brain, just going shh. Hush, little neuron. I don't know how to remember that song. It's a shush. It's the shush transmitter. It's the shush transmitter. Everywhere in the neuron. Very common biological system. Okay. If I am a neuron that makes GABA, I also have an enzyme that I produce. I have the gene sequence for glutamic acid decarboxylase. What does a decarboxylase do? Carboxyl, okay? Which is that dude up there? Nope, not gonna get it. This is the carboxyl group that gets cut by that, produce GABA. So remove 2 oxygen to hydrogen and a carbon, boom, now glutamate becomes GABA. Completely opposite function. Again, do not know structure. Know your enzymes there. So let me ask you this: if I make GABA, do I also? Well, no, do I also produce the enzyme glutaminase? That's a tough question. If I make GABA, I need glutamate to be made first. But I don't release glutamate, I just use glutamate to make GABA. Does that make sense? So if I make GABA, I have to have everything that the glutamate releasing neuron has. And then I have one more thing, which is glutamic acid decarboxylase. So you need glutaminase to make the glutamate, so you can make GABA from the glutamate. You're going to get a question like that on exam too. It's so important. So if I make GABA, I also transport glutamine up into my neuron, into my synapse, or into my nerve. So it looks exactly like the glutamine neuron, or glutamate neuron, producing neuron, but has the extra step of glutamic acid decarboxylase, chopping off that COOH off of glutamate. Boom, now I'm GABA. Yep.
GABAergic neurons should never interact with glutamine itself, right?
Sure they would, because they need the glutamine. The question was GABAergic neurons would not interact with glutamine. But yeah, they need the glutamine to be uptaken to make the glutamate, which is then used to make GABA. I mean, it's complicated, sure. Other questions? It's definitely complicated. 100%, I'm on board, I get it, okay? Just like with glutamate, nearly all GABA is is uptaken after synaptic release by a nearby astrocyte. Because GABA, just like glutamate, is a substrate in the Krebs cycle, and if you reuptake 100% of GABA and it's free floating in the cytosol, you're going to mess up energy production in the Krebs cycle. I'm not going to ask you any questions about the Krebs cycle. So astrocytes that reuptake GABA have a GABA transporter. GABA gets released by a neuron, inhibits a receptor. Free GABA gets sucked up out of the synapse by a neuroastrocyte through the GAT or the GABA transporter. The third most commonly prescribed medication for seizure disorders does something related to this slide. Want to take a shot? How would interacting with this slide in particular help to reduce seizure activity? Or migraines? Works for that too. Or chronic pain disorders, which are overactivity of axons that bring pain signals into the spinal cord and brain. That's the one people go, gabapentin was supposed to do this, but it really doesn't. What happens if I inhibit GABA transport up into an astrocyte after it's been released by a neuron? Continues to shush further neuronal activity. Seizure disorders are overactivity of neurons. Third most commonly prescribed medication to treat seizure disorders inhibits GABA uptake into astrocytes. That's why this stuff is important for neuropharmacology. Yeah? Can that be dangerous in largemouth? Yes. Yes. You'll always need a moderate level of glutamate and GABA activity. Now, if we make GABA too active, is that a person, a cell phone, a dog? Somebody's messing around with us. Okay. So one of the things alcohol does is overstimulates GABA receptors to reduce action potential. You can do the same thing with an anti-seizure medication. So dosing is very important. Okay? There's your GABA receptor incomplete. Questions? Yeah. Oh. Yeah. Oh. Yes. On the neuronal side, the only difference is glutamic acid decarboxylase, or GAD. So I'm not going to ask you medication names. That starts after exam 2. Just an example of FDA-approved prescription medications to treat seizure disorders, migraines, and chronic pain conditions. Activating GABA receptors to allow chloride in, blocking AMPA receptors. Depakote inhibits GABA breakdown, but you get the point. If we reduce sodium channels from being active, and we increase GABA receptors being active, we can control seizures fairly well. Not for everybody, but for most people. That's why the mechanism is so important, because that's where we start when we're developing new drugs. We start by looking at diagrams like this. Well, what could we do to GABA uptake efforts release to help with seizure disorders, migraines, or chronic pain? Questions? All right, have a good weekend. I will see you Tuesday for exam review only. Bug me tomorrow in an email if I forget to make the top hat quiz questions available.